If you have Rh-negative blood, there's a piece of your genetic code that has puzzled scientists for more than 80 years. And even now, with every tool modern genetics has to offer, they still cannot fully explain how it survived. Not why it exists, why it survived. Because by every rule evolution is supposed to follow, this trait should have vanished from the human species almost as soon as it appeared. It didn't. Today it runs through the veins of hundreds of millions of people, clustering in patterns across the globe that still do not fully line up with how humans are known to have moved, mixed, and settled the planet. This is the story of where that blood actually comes from, what it cost the people who carried it, and why, against every evolutionary expectation, it is still here. The story starts not in ancient history, but in a laboratory in New York City in 1940. Carl Landsteiner, the same physician who had already identified the A, B, and O blood groups 4 decades earlier, was working alongside a younger immunologist named Alexander Wiener. Together, they were investigating how the immune system reacts to unfamiliar proteins, and their experiment involved injecting rabbits and guinea pigs with blood cells taken from rhesus macaque monkeys. The animals' immune systems responded as expected, producing antibodies against the foreign monkey cells. But when Landsteiner and Wiener took those antibodies and mixed them with human blood samples, something odd happened. The antibodies reacted violently with roughly 85% of the human samples, clumping the blood cells together in a process called agglutination. The remaining 15% showed no reaction at all. But that reaction, or the absence of it, revealed a protein sitting on the surface of most human red blood cells, a protein that a smaller portion of the population simply did not have. Because the discovery came through experiments involving rhesus monkeys, Landsteiner and Wiener named it the rhesus factor, later shortened to Rh. People carrying the protein were classified Rh positive. People without it were Rh negative. It seemed, at first, like a minor addition to blood typing, useful mainly for improving the safety of transfusions. It would soon prove to be much more than that. Doctors began noticing a disturbing pattern in maternity wards that no one could fully explain. Infants were being born with severe anemia, dangerous jaundice, swelling, and, in the worst cases, stillbirth. The condition had a name, hemolytic disease of the newborn, but for years its cause remained a mystery. The Rh discovery finally supplied an answer. When an Rh-negative mother carries an Rh-positive fetus, a small amount of the baby's blood can cross into the mother's bloodstream, usually during delivery. Because her immune system has never encountered the Rh protein before, it may begin producing antibodies against it, treating the baby's blood as a foreign threat. In a first pregnancy, this is often harmless, since the exposure and the antibody response happen too late to affect that child. But in subsequent pregnancies with another Rh-positive baby, those antibodies are already present and ready to attack, and the consequences can be severe. Before treatment existed, this condition was devastating on a global scale. In the United States alone, researchers estimate that around 10,000 infants died from hemolytic disease of the newborn every year prior to the late 1960s. Physicians described it as a kind of biological tragedy playing out inside the womb, where a mother's own immune system, meant to protect her children, was instead being turned against them by an invisible mismatch in blood chemistry. For centuries before anyone understood the mechanism, families experienced this as an unexplainable pattern. A first child born healthy and subsequent children lost or gravely ill with no apparent cause. The breakthrough that changed this outcome took nearly 3 decades of additional research to arrive. In the 1960s, physicians including Vincent Frieda and John Gorman at Columbia University, working with immunologist William Pollack, discovered something almost paradoxical. The same anti-Rh antibodies responsible for causing the disease could, if administered to an Rh-negative mother shortly after delivery, prevent her immune system from mounting its own lasting response in the first place. Given at the right dose and within a narrow window after birth, this treatment could stop the sensitization process before it started. In 1968, the United States Food and Drug Administration approved the resulting therapy, marketed under the name RhoGAM. A New Jersey mother named Marianne Cummins became one of the first recipients that year. Within just a few years, tens of thousands of infant deaths that would once have been considered routine and unavoidable were being prevented annually. It remains one of the most effective and least publicly known achievements in 20th century medicine, but even as the medical danger of Rh-negative blood became clear, a separate and far stranger puzzle was taking shape, one that had nothing to do with treatment and everything to do with origin. Roughly 15% of people worldwide carry Rh-negative blood, most commonly caused by a complete deletion of the RhD gene on chromosome 1, the gene responsible for producing the Rh protein in the first place. In genetic terms, this is not damage or disease. It is simply an absence, a clean deletion that switches the protein off entirely. And that absence is not evenly distributed across humanity. It clusters, sharply and unevenly, in specific populations around the world, in ways that continue to challenge simple explanations. Nowhere is that clustering more extreme than among the Basque people, a population living in the mountainous border region between northern Spain and southwestern France. While most European populations sit somewhere between 15 and 18% Rh negative, various studies of Basque populations have reported dramatically higher figures, with some historical samples measuring phenotype frequencies in the mid-30s% range, and more recent genomic research reporting underlying RHD deletion allele frequencies even higher than that. The exact numbers vary depending on whether researchers are measuring the visible blood type or the underlying gene frequency, which are related but not identical statistics, and that distinction matters, because much of the popular retelling of this story blurs the two together. What is not disputed is that the Basque population carries this trait at a rate that makes it a genuine outlier, unmatched by any other population on Earth. Researchers studying this discrepancy have pointed out that phenotype frequency, meaning the percentage of people who actually test as Rh negative, and underlying allele frequency, meaning how common the deletion variant is within the full gene pool, are related but mathematically distinct measurements, and popular articles online routinely blur the two together into a single, simplified number that overstates how settled the science actually is. The Basques are unusual in other ways too. Their language, Euskara, is what linguists call a language isolate, meaning it has no confirmed relationship to any other language family on the planet, Indo-European or otherwise. Genetic studies, including research associated with population geneticist David Reich and colleagues, have traced Basque ancestry back to early European hunter-gatherer and early farming populations that inhabited the Iberian Peninsula during and after the last ice age. When large-scale agricultural migrations swept into Europe from the Near East, beginning around 7,000 years ago, reshaping the genetic makeup of most of the continent, the population that would become the Basques appears to have remained comparatively isolated in the shelter of the Pyrenees, absorbing relatively little of that incoming ancestry compared to their neighbors. It's important to be precise here, because this fact has been stretched into some fairly wild claims online. The Basques are not a separate branch of humanity, and there is no credible genetic evidence supporting theories of a distinct or non-human lineage. Every chromosome, every haplogroup identified in Basque populations traces back to the same ancestral human populations found elsewhere in Ice Age and early Neolithic Europe. What makes the Basques distinctive is not a different origin, but a long period of relative genetic isolation which allowed specific traits, including a high frequency of the RHD deletion, to become concentrated through founder effects and genetic drift, rather than diluted through repeated mixing with incoming populations. In other words, the Basques may be less a biological anomaly and more a genetic time capsule, a population that preserved something closer to Ice Age Europe's original genetic profile, while the rest of the continent was being reshaped around them. The mystery deepens when you zoom out to the global map. In Western Europe generally, Rh negative frequencies run in the range of 15 to 18%. Certain Jewish and Arab populations in parts of Western Asia have been reported with frequencies reaching into the 20s. North African Berber populations also show elevated rates compared to the regions surrounding them. Then, moving east, the numbers fall sharply. Studies of Chinese and other East Asian populations have found Rh negative frequencies below 1%. Some estimates placing it closer to 0.4%. Sub-Saharan African populations likewise show very low frequencies, generally under 1% as well. This geographic pattern alone raised questions for decades, but the deepest puzzle isn't really about where the trait is common. It's about why it exists at any meaningful frequency at all. Natural selection is, by definition, unforgiving toward traits that reduce reproductive success. Hemolytic disease of the newborn was, for most of human history, exactly that kind of trait. A condition that could and did kill children before they had the chance to have children of their own. Basic evolutionary theory predicts that a genetic variant carrying this kind of cost should be gradually eliminated from a population over generations, especially in a population like the Basques, where the trait is common enough that Rh negative mothers would have regularly encountered Rh positive partners. And yet it persisted, and in some populations it didn't just persist, it thrived. This is the part of the story that has genuinely occupied population geneticists for decades. Not as clickbait, but as a real, published, still partially unresolved research question. A 2012 study led by researchers including Alyssa Ann Wilson examined genomic data from multiple human populations, specifically to test whether some unknown selective advantage might be offsetting the reproductive cost of the RHD deletion. Their conclusion was that the pattern of genetic variation surrounding the deletion was consistent with a history of positive selection, meaning something about carrying this deletion, at some point in human history, provided enough of a benefit to counteract its clear reproductive risk. What that benefit actually was remains to this day an open question in the scientific literature, not a settled fact. Several hypotheses have been proposed, and it's worth being honest about how solid each one currently is. One line of research has examined possible differences in how Rh negative and Rh positive individuals respond to infection by Toxoplasma gondii, a single-celled parasite that currently infects an estimated 1/3 of the global human population, most commonly transmitted through undercooked meat or contact with infected cat feces. Once inside a human host, the parasite is capable of crossing the blood-brain barrier and forming dormant cysts within brain tissue, a process that some research has linked to subtle changes in reaction time, risk-taking behavior, and reflex speed in infected individuals. Population living by hunting, gathering, and near-constant exposure to wild and domesticated animals, even a small shift in reaction time or risk perception could plausibly have carried real survival consequences over many generations. A handful of studies, led primarily by researchers investigating the Rh protein's broader role in cell membrane function, have suggested measurable behavioral or physiological differences between Rh negative and Rh positive individuals infected with the parasite, with some proposing that the Rh protein itself may be involved in how efficiently the parasite operates within a host cell. This research is real and published, but it remains limited in scope and sample size, and no broad scientific consensus currently exists connecting it directly to the historical spread of Rh negative blood across specific populations. It is a candidate explanation, actively studied, not a proven one. A second area of research has looked at disease resistance more broadly, examining whether Rh blood type correlated with survival during historical epidemics, such as the Black Death, which killed an estimated 75 to 200 million people across Eurasia in the 14th century. Some researchers have proposed that regions with elevated Rh negative frequency show patterns consistent with differential survival during major plague outbreaks, though establishing a direct causal link across 7 centuries of incomplete historical records is extraordinarily difficult. And this remains a hypothesis, under active discussion rather than an established finding. A third, much more recent line of research emerged during the COVID-19 pandemic, when several teams tried to determine whether ABO and Rh blood types correlated with infection severity. The results have been genuinely mixed, and it's worth walking through that honestly rather than picking the one finding that makes the best headline. A study out of Poland found that Rh negative individuals were more likely to have had an asymptomatic course of infection, and pointed to a similar pattern reported in a large Canadian cohort out of Ontario, where Rh negative patients showed a lower risk of severe disease or death, but other research groups, including a retrospective review from a hospital system in the United States, found the opposite pattern, with Rh negative patients showing worse outcomes and higher mortality within certain blood group combinations. Several additional studies found no statistically meaningful association between Rh status and COVID-19 severity at all. Taken together, this body of research doesn't currently support a confident claim in either direction. What it does support is the idea that scientists are still actively probing whether the Rh protein plays some functional role in how the immune system handles viral infection, a question with roots stretching back to earlier findings that Rh negative status may offer some protection against unrelated viruses, such as parvovirus B19, and that Rh negative men may show a stronger interferon gamma immune response to influenza A. None of this proves an evolutionary origin story on its own, but it does suggest the Rh protein may do more inside the immune system than blood banks have traditionally needed to care about. There's also a geographic wrinkle worth mentioning because it complicates any story that frames Rh negative blood as purely a European phenomenon. In many sub-Saharan African populations, the Rh negative phenotype is genuinely rare, but where it does appear, it is frequently caused by a different genetic mechanism than the clean RhD deletion common in Europeans. Instead of the gene being physically removed from the chromosome, a non-functional, silenced version of the RhD gene remains present but inactive, sometimes called a pseudogene. The outward result, no Rh protein on the cell surface, looks identical in a basic blood test. But the underlying genetic cause is different, which tells researchers that RhD negativity likely was not a single event that spread outward from one ancestral population. It appears to have arisen independently through different genetic roots in different human populations at different points in history. That detail alone undercuts the idea of one single dramatic origin story, alien or otherwise, and instead points towards something more genuinely interesting. Multiple separate genetic experiments run by evolution itself, arriving at a similar outcome. What all of this research points to, cautiously, is a hidden trade-off. Something about carrying this deleted gene appears to have offered a survival advantage significant enough in specific environments and specific historical moments to balance out the reproductive cost that came with it. Evolution rarely preserves a costly trait without some corresponding benefit. Scientists simply haven't fully identified what that benefit was, or whether it was even the same benefit in every population where the trait became common. One detail that deserves more scrutiny than it usually gets in popular retellings of this story is the age of the RHD deletion itself. Many online articles state a precise figure, often somewhere between 25,000 and 35,000 years ago, presenting it as settled fact. The genuine research picture is less tidy. Peer-reviewed studies on the molecular structure of the Rh gene locus describe the deletion as arising through a specific mechanism, an unequal crossover event between two nearly identical DNA segments flanking the RHD gene, sometimes called the rhesus boxes, which allowed the gene to be cleanly excised from the chromosome. But pinning down exactly when this happened and whether it happened once or independently on multiple occasions in different populations remains genuinely unresolved in the scientific literature, complicated by subsequent recombination events and the difficulty of dating a single mutation event using modern population data alone. What can be said with confidence is that the deletion is ancient, that it predates recorded history by a wide margin, and that its full evolutionary story is still being actively investigated rather than fully written. There's one more historical thread that's worth including, mainly because it shows how old this fascination with Rh-negative adjacent traits actually is, long before anyone had the tools to test blood at all. In medieval Spain, aristocratic families began describing themselves as having blue blood, a phrase that came from the visibly bluish veins seen through the pale, sun-shielded skin of the nobility, in contrast to the sun-darkened skin of laborers. It had nothing to do with actual blood chemistry and everything to do with class, sunlight exposure, and a culture of dynastic intermarriage designed to keep bloodlines and inherited wealth tightly closed. Some later writers have tried to connect this idea directly to Rh-negative genetics, given how European royal families intermarried across generations in ways that would have concentrated any rare recessive trait, Rh-negative status included. This connection is plausible in a general genetic sense, since prolonged inbreeding within closed dynasties does tend to concentrate rare alleles of all kinds, but there is no rigorous genetic study establishing that Rh-negative frequency specifically was elevated among historical European royalty compared to the general population. What is well documented is the flip side of that same intermarriage pattern. European royal lines, most famously the descendants of Queen Victoria, also concentrated harmful recessive conditions like hemophilia alongside a range of other hereditary health problems, as centuries of marrying within a small, closed gene pool eventually caught up with them. The same genetic mechanism that could concentrate an interesting trait could just as easily concentrate a dangerous one, and royal history shows unmistakable evidence of the second outcome, whatever role the first might have quietly played. This uncertainty hasn't stopped a flourishing ecosystem of speculation from filling the gap. You have probably encountered some version of it. Rh-negative blood has been claimed, without any credible scientific basis, to originate from extraterrestrial ancestry, with proponents pointing to the fact that Rh-negative mothers can reject Rh-positive fetuses as supposed evidence of a foreign, non-human bloodline. Others have tied it to lost civilizations, ancient sunken continents, or claims of hidden royal bloodlines descended from some exceptional ancestral population. None of this holds up under actual genetic scrutiny. Every population studied, including the Basques, carries the same 23 pairs of chromosomes, the same broad ancestral markers, and the same fundamental human genome as every other population on Earth. The RHD deletion is a well-characterized, well-understood genetic event. It is not evidence of a separate lineage. It is evidence of a specific mutation that happened to persist, likely because of some genuine, still partially mysterious, selective advantage, concentrated by geographic isolation in certain populations and diluted by migration and intermixing in others. If there is a more grounded version of the mystery, it lies not in ancient aliens or lost continents, but in something more geologically real. The dramatic reshaping of the world's coastlines at the end of the last ice age. As glaciers retreated roughly 12,000 years ago, global sea levels rose by more than 400 feet over subsequent millennia, permanently submerging vast stretches of previously habitable land, including large parts of what is now the North Sea and areas of Southeast Asia that were once dry land bridges. Populations living in those coastal regions would have been forced to relocate inland or to higher ground repeatedly over many generations. Some researchers, studying the geographic distribution of Rh-negative frequencies, have noted that many of today's hotspots, including parts of Atlantic-facing Europe and North Africa, sit near regions that experience this kind of repeated coastal displacement. This is a genuinely interesting area of ongoing research, but it should be presented as exactly that, an area of ongoing research and hypothesis, not a fully proven historical account. What is not speculative, and what deserves far more public attention than the fringe theories usually receive, is how directly this ancient genetics story continues to shape modern medicine. O-negative blood, the type lacking both the A and B antigens as well as the Rh protein, is considered the universal donor type because it can be transfused into patients of nearly any blood type without triggering an immune reaction, making it indispensable in emergencies where there is no time to determine a patient's actual blood type. But O-negative blood is also rare. It accounts for roughly 7% of the population in most estimates, and current reporting indicates that blood banks across the United States have faced serious, sometimes critical shortages of O-negative supply, with some regional blood centers reporting drops of over 30% in available stock during recent shortage periods. Fewer than 1 in 14 people who actually carry this blood type donate regularly, according to blood bank officials, which means the very trait that makes this blood so valuable in trauma medicine is also what makes it perpetually difficult to keep an adequate supply. The medical risk tied to Rh incompatibility itself hasn't disappeared either, even though an effective treatment has existed since 1968. Research published in the medical literature estimates that a substantial share, in some analyses roughly half, of Rh-negative mothers worldwide who need an Rh immune globulin treatment during pregnancy do not currently receive it, largely due to gaps in awareness, access, or affordability in various parts of the world. That gap translates into an ongoing, entirely preventable burden of fetal and neonatal illness and death in regions where this decades-old treatment remains out of reach. The mutation that geneticists are still working to fully explain isn't simply a historical curiosity. It is an active, ongoing medical reality for millions of families today. It's worth sitting with the scale of that contrast for a moment. A genetic deletion that likely first appeared somewhere in humanity's deep, undated prehistory, quietly carried down through hundreds of generations, is directly responsible both for one of the more remarkable open questions in modern population genetics and for a measurable, preventable share of infant mortality happening in hospitals right now in this decade. Few traits connect ancient evolutionary history to a present-day maternity ward this directly or this consequentially. So where does that leave the actual answer to where Rh-negative blood came from? Not with aliens, not with a lost continent, and not with a secret royal bloodline. It leaves us with a genuinely ancient mutation, a clean deletion of a single gene that appeared at some point in humanity's deep prehistory and then, against the basic mathematics of natural selection, refused to disappear. It concentrated most heavily in isolated populations like the Basques, who preserved it not because they are biologically different from the rest of humanity, but because history simply left them less mixed than almost anyone else on the continent. It very likely persisted because it offered some form of protection against disease, quite possibly more than one disease, across different populations in different centuries in ways researchers are still working to fully untangle. And it continues today, not as folklore, but as a measurable medically critical presence in emergency rooms and maternity wards around the world, carried quietly in the blood of roughly 1 in every 7 people alive. If this is the blood type running through your own veins, you're carrying a genuinely unresolved piece of human evolutionary history, one that professional researchers are still actively studying, not a settled mystery and definitely not a mystical one. That, honestly, is more interesting than any of the myths built around it. If you found this breakdown useful, subscribe for more deep dives into the real science behind the genetic stories that usually get buried under speculation. Drop your own blood type in the comments if you want to see how common your version of this story really is, and hit like if you'd like to see more videos separating verified research from internet mythology.